Good afternoon, everyone, and welcome to today's session. Um, we'll wait for 5 minutes so that people can join in, and then we're gonna begin our discussion. Not begin, actually, continue our discussion with respect to membrane proteins. Okay. guess we can start our discussion for today. So I'm gonna just summarize or maybe recap a little bit what we have studied in the last class, as we have started a new topic, that is membrane proteins, and we are done with membrane lipids. So as membrane proteins are the ones which provide the functionality associated with the membrane, right? Because lipids are majorly the structural part of the membrane, while proteins corresponds to the functionality and the functions which are primarily associated with the biological membrane. So in the last class we have actually discussed about amino acids, because at the end of the day what proteins are the polymers of amino acids, right? So proteins are polymers of amino acid, and each amino acid are joined together by a specific type of covalent bond called peptide bond, which we're gonna see in today's session. Okay, so each amino acid in a protein is called amino acid residue. Okay, and then we studied about their basic structure, what are the backbones of amino acid. Then we actually spent quite a lot of time in classifying the amino acids based on their side chain, or the R group or the variable group. So based upon the differences in the side chain or the R group, there are majorly 5 categories of amino acid. First category being non-polar and aliphatic. It contains 7 amino acid named glycine, alanine, proline, valine, leucine, isoleucine and methionine. Second group being aromatic amino acids, which have aromatic side chains. They are phenylalanine, tyrosine and tryptophan. Then we have polar or uncharged amino acids. They are serine, threonine, cysteine, asparagine and glutamine. Then we have positively and negatively charged amino acids. In positively charged amino acids we have lysine, arginine and histidine. In negatively charged amino acids we have only 2 negatively charged amino acids in the living system. They are aspartate and glutamate. Then we studied about the basic characteristics of these categories, that is non-polar aliphatic, aromatic, polar uncharged, positively charged and negatively charged. Okay, so to summarize our last week's session, we can summarize it in this way. The 20 amino acids commonly found as residues in protein contain an carboxylic group, an amino group and a distinct R group substituted on the alpha carbon. The carbon atom of all amino acids except glycine is asymmetric, and thus amino acid can exist in at least 2 stereoisoforms, L form and the D form. Okay, only the L stereomers of amino acid with a configuration related to the absolute configuration of the reference molecule L-glyceraldehyde are found in proteins. Amino acids can be classified into 5 types on the basis of polarity and charge at pH 7 of their R group. Other less common amino acids also occur either as constituents of protein, usually through modification of common amino acid residues, or as free metabolites. Amino acids vary in their acid-base properties and have characteristic titration curves. Monoamine monocarboxylic amino acids with non-ionizable R groups are diprotic acids at low pH and exist in several different ionic forms as the pH is increased. Amino acids with ionizable R groups have additional ionic species depending on the pH of the medium and that of the R group. These 2 points, they are majorly indicating towards the diverse properties of the amino acids and the proteins in general. The properties of a protein, the properties of proteins, where will it come from? It will come from the amino acids at the end of the day, right? And amino acids, their property varies depending upon the surrounding, indicating here that whether they are at high pH or low pH. Again, this high pH and low pH will correspond to what type of amino acids we are talking about. So if we go back to our basic classification of amino acids, so if we have hydrophobic amino acids, right, then at physiological pH either they will be protonated or non-protonated. Similarly, if you are talking about negatively charged amino acids, if the physiological pH is not according to these amino acids, then the properties of these amino acids will change depending upon the surrounding. Okay, so that was one type of classification of amino acids. As I have mentioned plenty of times in the last class, that you can actually classify amino acids in various groups. It will completely depend upon what criteria you are selecting to classify them, right? So it's very easy to understand. For example, let's take a very daily life example. If I say, for example, there is a classroom. Okay, so in classroom you can actually divide students in multiple ways. So the easiest way to classify the class will be into number of how many female students are there or how many male students are there, right? So you can classify the same class, suppose class A, in two categories, one which only has female students and the other which only has male students, right? Same class you can classify into multiple classes. For example, if I talk about CGPA, right, those people who have their CGPA from 8 to 10, they'll belong to one class. Then 5 to 8, they'll belong to next class, and then everyone below 5 CGPA will belong to another class. So same class you can either divide into two subclasses, or you can divide into three or multiple subclasses depending upon the criteria which you are selecting right now. So the criteria which we selected here, in which the amino acids were classified in 5 different groups, that criteria was based upon the side chain or the R group. Okay, so same 20 amino acids, same biologically relevant 20 amino acids can also be categorized into two classes if we look only at the polarity of the amino acids. So they can be non-polar amino acids and they can be polar amino acids. So in non-polar amino acids we have alanine, valine, leucine, isoleucine, proline, phenylalanine, tryptophan and methionine. Okay, if we talk about polar amino acids, then we have glycine, serine, threonine, cysteine, glutamine, tyrosine, asparagine, lysine, arginine, histidine, glutamic acid and aspartic acid. So this is how amino acids are written. The small 3-letter words is called the 3-letter code, and the single alphabet is a single representation of that particular amino acid. For example, if you see a FASTA sequence of a protein or any protein sequence, you will mostly see these single letter code of the amino acids. Okay, so A being alanine, F being phenylalanine, M being methionine, and that is how amino acids are represented in a protein structure. Now if we look at the other way how we can classify amino acids, is again similarly based upon the side chain or the properties of the side chain. They can be aliphatic if the side chain is of aliphatic nature, aromatic, polar uncharged, sulfur containing, cation, anion and imino. So aliphatic will be glycine, alanine, valine, leucine and isoleucine. Aromatic amino acids will be phenylalanine, tyrosine and tryptophan. Polar uncharged will be serine, threonine, asparagine and glutamine. Sulfur containing will be cysteine and methionine. Only 2 sulfur containing. This is important to remember, that in the biological system only 2 amino acids have sulfur in them, cysteine and methionine. Out of cysteine and methionine, only cysteine is the residue which makes disulfide bond. Why? Because it has free SH group, while in methionine the sulfur is not free. Apologies for actually going back and forth, but it is important to look here at the structure of methionine and cysteine. So if you look at methionine and cysteine, both of them have 1 sulfur in them, right? But this sulfur, it's not free to make any bonds with the next methionine entity. On the other hand, this cysteine, this can actually make another disulfide bond with the cysteine. So if you remember, cysteine means this single cysteine amino acid residue, while cystine, cysteine molecule, is the one in which two cysteine residues are attached with a disulfide bond. I guess I have mentioned it somewhere in the literature. It's this one. See, cysteine is readily oxidized to form a covalently linked dimeric amino acid called cysteine. So it's a very critical wordplay. So in most of the exams, this is a very direct question. People can ask directly that what is the difference between cysteine and cysteine. Okay, so cysteine is a single amino acid residue, while cysteine is the one which has two cysteine residue attached by a disulfide bond. Okay. Then coming to the next part, cationic amino acid residues will be the amino acid whose side chain are cationic in nature will be lysine, histidine and arginine. Anionic one will be aspartic acid and glutamic acid. Amino acid will be proline because it has that amino side chain. You guys can refer back to the previous lecture where I have actually discussed about the structures of amino acid. Okay. So from this, the overall objective was for you guys to understand that it is not necessary that whatever the classification I am teaching you, these are the only ways you can classify amino acids. The moment you change your categorizing criteria, n number of classes can be there. So if someone asks you a question, how many different types of amino acids are there, you cannot answer this question, right? Because they haven't mentioned on what basis. It can be on the base of polar or non-polar, then the answer will be 2. If it will be based upon the chemical nature of the side chain, then it will be 7. If it will be based upon the type of side group or the R group, then it will be 5. So it is very important to mention while answering such questions that on what basis the classification is being asked. Okay. So till now we have discussed about the amino acids. So amino acids are what? They are the monomers, correct? And what are proteins? Proteins are the polymers of amino acid. So how these polymers are actually formed, how they are linked together. So two amino acid molecules can be covalently joined through a substituted amide linkage termed as peptide bond to yield a dipeptide. Such a linkage is formed by removal of the elements of water, a hydroxyl moiety from the carboxylic group of one amino acid, and a hydrogen atom, the amino group of the another amino acid. So you can see here, this is one amino acid, like R1 indicates one type of amino acid and R2 indicates another type of amino acid. The backbone remains same, NCC, right? So this is the backbone of all the amino acids which we have, only the side chain changes. So the carboxylic end of first amino acid residue, it has OH associated with it, right? And the amine group of second amino acid, it has one H associated with it. So once they are brought together in the reaction within the reaction distance, then there is a dehydration mechanism undergoes and this water is eliminated. Okay, and water is eliminated, making a NCC bond or CCN bond. So this bond, this carboxylic group of first amino acid attached to the amine side of the second amino acid, this particular bond is called, this particular bond is called peptide bond. Okay, and this is a very unique type of bond, though it looks like a single bond, right? Because it's a covalent bond and it's a single bond at the end of the day, formed by the dehydration mechanism, correct? But if you look at basic small molecules, small chemical compounds which make such bonds after dehydration, they are single bond in nature, but the energy associated with this peptide bond and the energy associated with the general covalent bond, there is a huge difference. The energy which is associated with the peptide bond is in between that of covalent bond of single covalent bond and double covalent bond, right? So this particular bond has energy in between that of single covalent bond and double covalent bond, indicating that this peptide bond is usually very stable at physiological conditions, because it is not a single bond. In general, covalent bonds are the strongest bond, right? But even in the covalent bond, single bond, double bond and triple bond. So triple bonds are the one which have very high energy associated with it, and hence it is very difficult for us at the physiological level, even for the enzymes, to break triple bonds, right? On the other hand, if we look at a peptide bond, the energy associated with the peptide bond is intermediate that of single bond and double bond, indicating that proteins are relatively more stable in comparison to molecules where there are single bonds present. Okay, when a few amino acids are joined in this fashion, the structure is called an oligopeptide. In this fashion means, after dehydration reaction, carboxylic end of first amino acid, amine side of another amino acid, dehydration reaction takes place, and that structure is called oligopeptide. When many amino acids are joined, the product is called a polypeptide. These are just very generic terminologies associated with the protein. It is important to know because these are very common terminologies in the field, and it will help you in actually communicating your things very effectively. Proteins may have thousands of amino acid residues. Although the term protein and polypeptide are sometimes usually used interchangeably, molecules referred to as polypeptides generally have molecular weight below 10,000 Daltons, and those called proteins have higher molecular weight. No generalization can be made about the molecular weight of biologically active peptides and proteins in relation to their functions. So this is kind of important. It's kind of easy and difficult at the same time to understand. So what this statement indicates, it says that no generalizations can be made about the molecular weight of biologically active peptides and proteins in relation to their function. Meaning, you cannot say that if the protein complex is very large, right, then it will do more function or more important function. So you cannot actually relate the size of the protein with respect to its function. There can be small proteins which might be working or which has functions associated with them which are very important for the survival of the cell or the organism. On the other hand, both peptides and proteins, these are the terms which are interchangeably used, but for you guys, a basic classification between peptide and proteins will be any peptide which has more than 100 residues will be termed as protein. Proteins will be peptides at the end of the day, but this is a very it's not a thumb rule, but a very general understanding in the field that if any protein is more than 100 residue long, then it will be called as a protein and not a peptide anymore. So to make my point more clear, you can look at some of the distribution of molecular weight and the number of residues and the polypeptide chains associated with some of the proteins. So if you look at cytochrome C of human, it has molecular weight of 12,400 Dalton, while the number of residues in cytochrome C is 104. And how many polypeptide chains or in how many different protein polymers are associated with this protein? It's only one, which means there will be one long thread-like structure having 104 residues which will comprise cytochrome C protein. Then myoglobin. Myoglobin has 16,700 molecular weight and residues are 153, and again it has single polypeptide chain. So you can see even a 104 amino acid residue long can be in single polypeptide chain. And similarly 153 residues, 153 is definitely more than 104, right? So even 153 amino acid residues can also form a functional protein just by having single polypeptide chain. Another one will be chymotrypsin. So if you look at the chymotrypsin, the weight is 25,200 Dalton. The residues are 241, and these 241 residues will be repeated 3 times, which means they will be multimeric protein. Correct? Multimeric, oligomeric. These are the terms we use when there are more than one polypeptide chain associated with the protein. Similarly, you can see for hemoglobin, which has 4 chains, hexokinase, which has 2 polypeptide chains, and imagine they have 972 amino acid residues for hexokinase, right? Definitely more than chymotrypsin. Chymotrypsin has 241, yet they have 3 polypeptide chain, while hexokinase, it has 972 residues and they are divided in only like 2 peptide chains. So by this we can actually say that the molecular weight and the number of peptide chains as well as their function, it is not at all related to their molecular weight. Okay. Excuse me. So amino acids can be joined covalently. Wait, let me get the laser pointer. Amino acids can be joined covalently through peptide bonds to form peptides and proteins. Generally, cells generally contain thousands of different proteins, each with a different biological activity, obviously. The ionization behavior of peptides reflect their ionizable side chain as well as the terminal amino and carboxylic groups. Proteins can be very long polypeptide chains of 100 to excuse me, I just need to drink a little bit of water, I'm sorry. Yeah. So protein can be very long polypeptide chains of 100 to several thousand amino acid residues. However, excuse me. However, some naturally occurring peptides have only a few amino acid residues. Some proteins are composed of several non-covalently associated polypeptide chains called subunits. Simple proteins yield only amino acid on hydrolysis. Conjugated proteins contain in addition some other components such as metal or organic prosthetic groups. We are not gonna dig more into this because I'm planning to actually have a separate lecture for protein structures and how the structures are determined. So it's another module, but before going to that module, I will actually describe basic protein structures. You guys might be aware of it, like primary, secondary, tertiary, all those, right? So I'm planning to have maybe 2 or 3 dedicated class only for the classification and the types of protein secondary structures, protein structures, not only secondary structures. So sorry for my coughing. Yeah, I'll try to speak more slowly and okayish now. Yeah. So as I've told you that I'm already planning to have a separate lecture for the structure of protein. So we're not gonna dig more into this because this information can easily be covered in those lectures. Okay. So in a protein's 3-dimensional structure, the more hydrophobic amino acids should reside together forcing that portion of the protein into the membrane, while the more hydrophilic amino acid should locate into protein's domain that will interact favorably with water. So this principle is similar to what we have seen in the lipids, right? So lipids were amphipathic in nature with polar head group and hydrophobic tail. So if you remember our like previous discussions with respect to amphipathic lipids, the polar head groups, they want to stay in polar environment which is provided by the water, while the long acyl chain, they are hydrophobic in nature and they want to stay away from the water, right? That is why how the lipid bilayer is formed, all the polar heads are facing towards the water interface and all the tails are facing towards each other, and those tails which are facing towards each other, they are forming that hydrophobic region, right? Similar principle is followed here also. So the amino acids which are hydrophobic in nature, they will definitely prefer to stay away from the water, right? So how this can be achieved in a protein? This can be achieved in a protein. For example, let's take a sphere, correct? So if a sphere is there, the outside or the surface of the sphere is exposed to air. Imagine it's in the water, okay? So the outside surface is in contact with the water. Hence, the amino acids which are present on the surface of this sphere will be those amino acid residues which are hydrophilic in nature, because they want to stay in water, they want to be connected with the water, right? While the residues which are at the core of the sphere or inside of the sphere, they are untouched by water because they enjoy hydrophobic environment, right? So in such way, proteins are assuring that all the hydrophilic molecules or all the hydrophilic amino acid residues are towards the water phase, and all the hydrophobic residues are in the hydrophobic region only, okay? Excuse me. Therefore, membrane proteins analogous to membrane lipids must also be amphipathic, having both hydrophobic and hydrophilic regions. It's pretty understood, right? The outstanding question is how the amino acid sequence relate to a protein's location either into or out of the membrane, right? It's very easy to say ki, okay, if the amino acid residue is hydrophilic, it will try to stay in water. If the residue is hydrophobic, it will try to stay away from the water. But how can we say that what sequence of amino acid is facing where, either inside of the cell or outside of the cell or in the membrane, right? So one direct possibility, and how we can be 100% sure that what residues are facing towards the cytosolic side or the extracellular side, which is hydrophilic in nature, or the membrane itself, which is hydrophobic in nature. So one direct possibility would be to determine the precise location of each amino acid in a protein by X-ray crystallography, right? This will be the ultimate proof. If you have precise location of every amino acid of a protein via X-ray crystallography, you don't need anything else, correct? Because you have the direct proof. You are literally viewing all the amino acids which are spanning the membrane, which are present in the membrane. But it is not that easy. Unfortunately, this has proven to be a very difficult task for most membrane proteins, and I'm going to discuss this maybe in the next class, why studying membrane proteins is a huge task even today. So there are now more than 30,000 known water soluble globular proteins and more than 20,000 membrane proteins. However, as it is much easier to obtain crystals from globular proteins, more than 6,000 crystallography structures of these proteins have been determined compared to a few dozen structures of integral membrane protein. You can see there's a huge difference, right? 6,000 crystal structures of globular proteins. Globular proteins are the proteins which are present in the cytosol. They are not associated with the membrane in any way. So we have around more than 6,000 such crystal structures. While we have only handful of crystal structures for integral membrane protein. And don't worry, we're going to discuss about integral membrane protein in the coming slides. So are there other methods to identify protein domains that prefer a location in the hydrophobic core of a membrane? So what is our objective? Our objective is to know that via protein sequence we don't know which protein will be outside, inside, or inside the cell. Will be facing outside the cell, or will be present inside the cell associated with the membrane, or will be in the membrane, right? There are three possibilities. So if you remember our basic fluid mosaic model, there were different types of proteins, right? Peripheral, integral membrane, membrane associated, all those. So is there a way, because we know there is a chemical property called hydrophobic residues will tend to stay in hydrophobic region. Lipid bilayer, the interior of a lipid bilayer is hydrophobic in nature. So can we relate the sequence of an amino acid with that of its span through the membrane, right? So the question is, can we do this? So to answer this question, the amino acid cannot simply be lumped into classes, but instead must be characterized individually. This approach involves hydropathy plots. So what it means, for example, if we are saying I'm sorry, I'm going back and forth again. So if I am saying that all these non-polar amino acids are there, right? So all these non-polar amino acids will like to stay in a non-polar environment. In the membrane, the non-polar environment is provided by the interior of a membrane. So if we generalize this, that all these amino acids will always stay in the membrane, this won't be a fair call. Why? Because there are obviously certain discrepancies associated with it, right? So the other way will be to look at each amino acid and the energy associated with that amino acid, whether they prefer to stay in water or they prefer to stay in non-polar media. So by this method, each amino acid is assigned a unique hydropathy index value that expresses its lipid water partition, indicating that how much energy is associated in bringing a amino acid residue from a hydrophilic environment to the hydrophobic environment. If amino acid wants to stay in hydrophilic environment, more energy needs to be supplied for it to bring to the hydrophobic part of the partition, right? On the other hand, if the residue wants to stay in hydrophobic environment, then less energy needs to be provided, or it will be a spontaneous process that it will come to the hydrophobic side. Okay, so these values represent a measure of the free energy change accompanying the movement of an amino acid from a non-polar solvent to water. Probably the most used approach for this type of membrane study came from Kite and Doolittle in 1982. So what they did is they actually calculated single amino acid residue and the free energy associated with them when that amino acid is being present in water and then when it is being brought to a non-polar solvent. So you can see this is the representative hydropathy indexes of all the 20 amino acids according to Kite and Doolittle. So by Kite and Doolittle, most of the non-polar amino acids have a positive index, while most of the polar amino acids have a negative index. Okay, so you look at here isoleucine, valine, leucine, phenylalanine, cysteine, methionine and alanine. Till alanine, all the values are positive, indicating that they are hydrophilic in nature. Okay, while glycine, threonine, tryptophan, serine, tyrosine, proline, histidine, glutamic acid, glutamine, aspartic acid, asparagine, lysine and arginine, all these are the ones which are hydrophobic in nature because the values associated with them according to the hydropathy index is negative. So from this we can somehow get an idea that in a protein sequence, if we see a stretch of hydrophobic residues, and the hydrophobic residues are ranging from glycine till arginine. Okay, so if we see such residues sorry, from glycine to arginine are the non-polar and hydrophobic amino acids. Yes, I am correct. Right, so we can somehow say that if the hydropathy index, or in a protein sequence, if there is a stretch of amino acids which predominantly belong to this part of the hydropathy index profile, we can say that these are the residues which prefer to stay in non-polar environment, right? I'm sorry, I guess I'm mixing something. Wait, just give me 1 minute. These are non-polar and these are polar. Okay, I guess I'm saying things vice versa. The things are vice versa. Okay, so let me repeat and correct myself. I am sorry for this. So isoleucine till alanine, these are non-polar amino acids, meaning they are hydrophobic in nature, indicating that if such residues are present in a single stretch in a sequence, if you look at the sequence, they might indicate that they want to stay in hydrophobic environment, and hydrophobic environment is provided by the interior of a membrane, correct? So we can somehow say, or somehow get an indication that if I have such residues in my protein sequence and they are continuously present, then that part of a protein will lie inside the membrane, inside the hydrophobic environment of a membrane. Okay, if you look at one such hydropathy profile, so this is how these hydropathic plots are formed. So once the hydropathy index for the amino acid is established, the next step is to plot the index values for each amino acid in the protein sequence from N terminal to C terminal. So I'll give you guys 1 minute. Okay, so if the hydropathy index, this is positive hydropathy index, positive values indicates non-polar nature. Okay, negative values indicate polar nature. Non-polar or hydrophobic residues will tend to stay inside the membrane, while hydrophilic residues will tend to stay outside the membrane. So looking at this graph, this is a hydropathy plot of glycophorin protein. Okay, looking at this graph, can you guys tell me which, and these are the sequence number residue, for example 20th residue is this, 40th residue is this, so this is a stretch, this is one stretch. So can you guys guess which part of this protein from the residue number or sequence number will likely to stay in the membrane? Because it should have a positive hydropathy index, it shows non-polar nature of the residues, and it indicates that it will be in the membrane. So can you guys tell me which part of the protein will be in the membrane? If we look at the residues ranging from first residue till 125th, 130th residue. Yeah. I hope you guys have a little bit of you guys might got the right answer. The answer is, if you look at the, so what I have asked you, I have asked that I want to know, I have a protein sequence, I have the hydropathy values of each residue, so I'll assign the hydropathy values of each residue to the sequence which I have, and then I will plot them. If the values are positive, indicating that those residues are hydrophobic in nature, and hydrophobic residues will tend to stay inside the membrane. So if you look at this graph, this huge patch is there, right, which has positive values associated with it. So this patch is roughly from maybe 75th, 73rd or 74th residue, right? From 75th residue till maybe 98, 99 maybe, right? Let's take 95 or something. So from 74 to 95, there is a large hydrophobic patch, indicating that this section of the protein will tend to stay in the membrane. So later when it was observed, this is the representative diagram of glycophorin protein, for which you guys are seeing this hydropathy plot. So if you look here, this is the 74th residue, this is the 95th residue. So from 74th to 95th, this huge stretch is actually in between the membrane and transversing the membrane. All other part of the protein is outside the membrane. Either it can face the cytosolic side or it can face the extracellular side. We are not concerned about this, but what we are concerned, and with this hydropathy index, so earlier there was no relation between the sequence and the location of a protein, right? But with Kite and Doolittle, or basically via hydropathy index, now we actually have somewhat idea that if a serial stretch is present of strong hydrophobic residues, we can say, if the structure is not available at the end of the day, right, then we can have a little bit of idea that this stretch will be facing the interior of a membrane, or will be transversing the membrane also. Okay, so this was an important finding at the end of the day. So to understand more about hydropathy index, it's a very small, like 3-minute video, which explains you and helps you in actually decoding the hydropathy profiles. So let me show you guys.
Step into Wix Studio, the platform for agencies and enterprises, and turn website layouts into an art form.
Hello everyone, and welcome back to the Biochemistry SI Program YouTube channel. My name is Jess, and I'm one of your content creators this semester. So today we're going to talk about all things hydropathy plots, and specifically what you need in order to solve a problem that you might encounter on an exam. So what exactly is a hydropathy plot? Well, a hydropathy plot is going to measure our hydropathy index, and it's going to measure the number of amino acid residues in a singular protein. What a hydropathy index does is it measures our free energy change associated with the movement of amino acid side chains from a hydrophobic environment to a hydrophilic environment. So when you see these graphs, you should immediately start to think of transmembrane protein. This is what a transmembrane protein looks like. Notice how it spans across the entire membrane. The amino acids inside the membrane are going to be hydrophobic, and the amino acids found outside of the membrane are going to be hydrophilic. So it's going to take about 20 amino acids in order to cross our membrane. So let's think about how you would solve a problem like this. First, we're going to need to know how to read our hydropathy plot. Make sure we understand that our positive free energy values are going to be associated with our hydrophobic amino acids, aka our amino acids found inside of the membrane. These are our negative hydropathy values. These are going to be associated with our hydrophilic amino acids, and they'll be found outside of our membrane. Let's take a look at an example of a protein. Notice how this protein crosses our membrane once, so it's a transmembrane protein. Since it crosses once, we can see that our hydropathy plot would look something like this. Notice how there is one positive peak. Again, notice how it crosses once. Let's say that our N-terminus is located on the outside or extracellular membrane or luminal face of the membrane. These are all different ways to say outside of the membrane. We would know that our C-terminus would have to be found on the inner membrane, or you can hear the words inner leaflet of the membrane. These mean the same thing. So let's look at a hydropathy plot. Notice how there are 4 positive peaks in our graph. So we're looking at a protein that's going to cross our membrane 4 different times. Now let's consider a question that could be asked based on this hydropathy plot. Consider the hydropathy plot for a transmembrane protein with its amino terminus located on the inner leaflet of the plasma membrane. Where is its carboxy terminus located? So let's walk through how I would solve this problem. So I'm going to set up my hydropathy plot here based on the question that was asked. I was told that the amino terminus is on the inner leaflet, so I will draw it like so. Now I know for each positive spike in my graph, I am crossing the membrane to another side. So for my first cross, if I start on the inside, I am going to end up on the outside. For my second cross, I come back into the inside. So for my third cross, we head back to the outer leaflet, and for the fourth cross, we know that our carboxy terminus must be in the inner leaflet of the membrane since our protein ends. If that didn't make sense, take a look at this protein and notice the 4 transmembrane segments. This protein is exactly what we just demonstrated from our hydropathy plot, so make sure that this makes sense to you, and if not, try to go back and redo the question. I encourage you to quiz yourself on these problems to make sure that you aren't just blatantly following along. For example, you should be able to answer the question, what would happen if our protein had only 3 transmembrane segments? Or what would happen if our carboxy terminus
right? So I hope that was pretty easy to understand, right? Similar thing you guys can apply if there are multi pass proteins, and don't worry about these terms. We're gonna actually have planned for the like different types of membrane, but it's okay, we'll cover this in the next class. So I hope that the class was fine and you guys were able to understand the importance of the relation of sequence and how we can relate it to the position of protein by looking at the sequence itself. It's a huge deal. Okay, so that's all for today from my side. If you guys have any question, feel free to drop me a mail, and the classes are as regular, Tuesdays and Saturdays. If there are any discrepancies in the schedule, I'll definitely inform you guys, but till now I guess no classes have been postponed except one Saturday class, which I'm planning to take maybe this week or next week as a extra class. Okay, so thank you so much guys for your attention, and bye bye.